So today I'm going to be making an interactive presentation using the Molecule Viewer. I'm going to specifically be using a magnesium channel just as an example. So this is a bacterial magnesium channel that regulates magnesium uptake. So what we're going to do is we're going to have the viewer open here, and we're going to go under File and put in the PDB ID, which in my case is 4I0U. Wait a couple of seconds, great. Okay, so just to get a clear look at this, go under Ribbon and select Chain. So I'm coloring by chain here, so you can get a better look at this. So you can see it's a dimer of pentamers. That's just how it crystallizes. So we just want to get rid of one of the pentamers. So what we're going to do is under here, go under Assembly 1. Great. So that leaves us with here. So our pentameric ion channel core A. So here's the profile of it. This is the membrane part right here. The ions travel through the pore and bind to the regulatory sites on the inside to close the channel. So what I'm going to do here is I'm going to make a couple of annotations. So right here, I'll call this the selectivity filter. I'll get to that in a minute. And here I'll call this the regulatory domain. Then deselect annotations, and I'll take a snapshot and call it, let's say, channel profile. Good. All right. So then what I'll do is, the function of taking a snapshot is it saves the current state. So what I'll do then, so now that's saved, I will get rid of these annotations really quick. Oops. Right there. Go under here. So you see there, I'm just looking for a magnesium ion right there. So so remember to deselect annotation before you do that. Okay. So click on one of the magnesium ions and go CPK so you can show a nice sphere of it. There it is. And what I'm going to do is click on atom type again. So you can see it's selecting the individual magnesium ions because I didn't want to fish for them one by one, so you can see that they're all there. And once again, click CPK and it brings up the rest of them. So you can see there's 10 of them here and an 11th one right there. So what I'm going to do, again, is click on it, again, under atom type, so it'll select all the magnesium ions. And we're going to color by, or rather, we're going to do a custom color, just because I'm not crazy about the green. So we'll make it some kind of a bright yellow, because there's no yellow yet in the structure. Okay, so there you have it. There are your magnesium ions in the regulatory domain. So one thing I might do, just to get a closer look, or just to show you an option here, we have a cutting tool. So what I can do is, zoom out here. We could spin it around, just see how that looks. Oops, it's a little bit, maybe a bit better. Yeah, something like that. Just to see how our channel looks like that. Yeah, so you see you can get a clear look at the domain. Although just for the sake of this, I might not use it. So just leave it out. But yeah, here you go. So I'll take I'll maybe make a caption of this at the top and say the regulatory domain of core A. And take another snapshot right there. And and deselect caption. Right, and call this regulatory domain. Regulatory domain, perfect. Okay, so the last thing I want to do is go to the top of the protein here and select a few of the highly conserved residues in the ion binding motif. So we have here, so sort of important to note is that when you're hovering over a part of the protein, it shows up in this corner right here, you can see, which is kind of nice. You can see here, that's how I found it so quickly. There's glycine 312, and I'm holding command now and selecting multiple residues, so methionine 313 and asparagine 314. And I'm going to do that for each of the chains. Just takes a couple of seconds. There's the methionine, the glycine, asparagine, glycine, methionine, asparagine. Great. So what I'm going to do now is show sticks for these residues and color the sticks by chain. And again, just as my preference is to show the oxygen as red, the nitrogen as blue, and the sulfurs as yellow. So I go under atom type and I just find one of each of these elements. It's pretty straightforward. And color by atom. Great. So just, again, just a preference. So here you have it. Oh, and I go back to residue here, and I'm going to double click on the magnesium ion just to set the pivot point to this. So that you can see now when I rotate, I'll rotate around the ion right there, so you can get a better look at the binding site here, how it's coordinated by these carbonyls behind glycine 312 and the asparagines right here. So one thing we can see here is that, let's see, so if you go into the measurement tool, just you can kind of start to also explore the structure and see if I made the realization that that the ion actually binds somewhat asymmetrically in this site, so that's kind of cool. But for now, what I'll do is just get rid of them, and deselect measure and just make a caption saying that. Oh, in this case, we'll change our caption here. Whoops. Change our caption and say something like Mg2 plus is coordinated by highly conserved GMN motif. And then deselect caption, and we'll take a snapshot and call this the selectivity filter. Great. So the idea here is that you can move between each of the snapshots. So you can see here the regulatory domain and the ion right there. Okay, great. So what we're going to do now is we're going to see how this looks under the presentation mode. So go under Share, press Get Link to Model, and update the thing. Sure, we didn't do anything, so that's fine. And there it is. So what we'll do is take this, and we'll just open it in a new tab. And here you have it. So you can just begin to look around the various parts of the protein in each of the snapshots here, and you can see the captions are right there. The annotation you saw before. Right? See it? It's kind of cool. You can explore it yourself. Yeah, there you have it. Presentation about magnesium uptake in bacteria using the molecule viewer.